introductions yet. Maybe we could do
I
a
was
round
just
of those.
thinking that.
Yes, thank you everyone. And so let me start with the introductions. My name is yyyy-mm-dd and I am the Science Communication Fellow for this watch.
Ali, good evening everyone. Daniel Wagner, watch lead.
Ali Mildo Tao, because it's already past midnight. Good morning, everyone. My name is Maikane Andres, and I'm the scientist on this watch.
Hello everyone, my name is May Lubetkin and I'm the science manager on the ship and the data logger on this watch.
In your front row, I'm Megan Putz, and I am your navigator.
Jake Bonney, I'm in the Herc seat.
I am Sarah Surgeon, and I'm in the Atalanta seat.
Got a shrimp in there.
Yeah, there's two different kinds.
And I'm Erin Rainey, and I'm in video engineer.
Just saw a nice shark on the surface.
Oh yeah,
Out back, yep.
wire
Oh,
cam.
good.
Swim around, yeah.
Hopefully Max saw it. He was looking for them earlier.
He really wants to see some.
Yeah. And I told him there would probably be some at midnight.
Can you send me another DVL reset,
Of course.
Megan? Thank you. What's this red?
Yeah. DVL doesn't like vertical cliffs. It's another anthomas, little mushroom coral.
Mushroom coral.
Yeah.
They're cute.
Anthomassus.
Hey Sarah, is this a good time to ask a question?
Sure.
So someone was wondering if Atlanta is there to help control the position of the tether on Hercules as to not get in the way, and do we always do ROV dives with a tether?
What was the first question?
Is Atlanta there to help control the position of the tether on Hercules?
Yeah, it's kind of like Atalanta. It's almost like an anchoring point, so it can take the ship's movement so that way Herc can kind of fly freely where it wants without having to get pulled in the direction or move with the ship so much. Helps stabilize it. And we do always go in with a tether. The tether or the cable is what provides power and communication to both vehicles.
Oh yeah, it's wild.
Thanks.
Yeah, I'll just add one thing to that last part. So yeah, we have remotely operated vehicles here on the ship, and they're all controlled by remote control via the tether, but there's also autonomous underwater vehicles which are not tethered, and we had earlier in American Samoa, the last two cruises, we had two autonomous underwater vehicles from Woods Hole Oceanographic Institution on board, AUV Sentry and AUV Mesobot, and so those did not have the tether. Now there's an issue there in that you got to recover the vehicle before you get the data, most of the data. Some of the data can be transmitted via acoustically, but yeah, there's a delay in getting most of the information back.
Thank you.
Oh, can we snap zoom on the black coral? It's right above that candelabra calyptrophora, that sort of grayish one.
Yep.
Keep an eye on the AC.
What's that? Paranthropathes? Or is it no?
Is it a paranthropathes?
No, it's a Bathypathes.
Go for zoom.
I wasn't sure. Yeah, I couldn't tell if it had more
beautiful.
branches. Like two different squat lobsters on it too.
Once in a while it flickers on.
Yeah, it's been going up and down.
So it looks like there's a sternostylus And a Europtychus squat lobster on there. You're a black coral person. Tell us about some black coral stuff.
Yeah, so this is going to be in the genus Bathypathis. Species will need to have a better look at it. So black corals are a group of corals most closely related to the hard corals or scleractinian corals. If you were to zoom in on those polyps, they would have 6 tentacles. They're unbranched. Black corals get their common name from the color of the skeleton. So now you're seeing the color of the tissue, which is brown or reddish. But black corals, most of them have quite bright colors. If you were to remove the tissue, the skeleton is dark black or brown in coloration. It's proteinaceous, so it's not made out of calcium carbonate as the hard corals are, so it's made out of protein. And yeah, they're quite interesting. The scientific name for this group is Antipatherans, derived from the Greek name antipathos, against disease or against evil. So in a lot of cultures, there have been amulets or medicines made from black corals to cure all kinds of ailments. In Hawaii, where I spend a lot of time, black corals have been used for a long time. So Native Hawaiians use those for medicinal purposes to cure lung diseases and mouth sores. But in many other cultures, there's been similar remedies. In Chinese culture. They're also used for jewelry. So the skeleton can be polished into quite beautifully. And so that's a species that's culturally and commercially quite valuable in many cultures. And they're also important ecologically, so they are habitat-forming species. As you can see there, the squat lobsters perching on top of them. If you were to zoom in, there's often also microscopic organisms that live on them, often worms and all kinds of things. There's a pretty interesting paper written a number of years ago where they harvested a single colony of a black coral and then just counted all the different animals that were on it, and it was in the order of thousands. A lot of little things. And
Oh.
Very cool. I've been checking out some of those like little sponges. They look like they might be polymastia, those little sponges on the rocks. All right, I think we got some great zoom on that. That was very cool. Atalanta's catching up to you, Jake.
Sweet.
Yeah, beautiful view here of all kinds of different organisms.
I love the Atalanta view. That's pretty wild.
It's a good shot.
Ooh, sea star.
Take a peek.
This is a cookie star of some kind.
There's a fish behind it too.
Oh, there is a fish. I think that's a macurrid, a rat tail fish. That's really cute. Jip is probably loving this.
Go for a zoom.
It could be something like Circeaster. Chris Ma would know. This group is particularly challenging for me. But that's why we have the Deep Sea Animal Guide. It's a collection of photos that we put together from dives just like this. And we've sent the images to taxonomic experts all around the world, and they've given us feedback on what these different animals are. And I use it almost every day to help me remember all the names. If you want to learn the names just like how I can spout them, that's how you learn. I was looking through the animal guide. At least that's what I do.
All right, come on.
Can we see if we can get that fish too?
Yeah, let's see that little fish.
There's one just below too.
It might be yeah.
But I think this might be a different one. But
I think it's the same one.
okay.
Might be Nezumia or something like that. Oh no.
There goes an inadvertent sample.
Shall we put it in the box, Daniel?
Yeah, let's do that if we're set up for it.
Set down right here.
Let's do it.
I'm just slowly coming up a little bit.
Well, you know, the Calyptrophora, we've been seeing that all over the place here. It's definitely one of the dominant species of coral that we've been seeing. So this will be a useful sample.
Yeah, absolutely. There's very, very sparse sampling from these depths here in Palau.
Hey, Daniel.
Yep.
So there is a question. A viewer is interested in asking if they are able to use this video footage for a creative project.
Absolutely. So the video is going to be distributed in different formats and in different time frames. So a big part of our mission for the Ocean Exploration Trust is to make data publicly available and accessible. So the video will end up on YouTube, both for scientific purposes and creative purposes. And all that we ask is that it's properly credited, making sure that we acknowledge our funders and the Ocean Exploration Trust. So for this expedition, NOAA's Office of Ocean Exploration and Research is providing the funding through the Ocean Exploration Cooperative Institute. There's also a request form on our website where people can request data, including digital data and video. So absolutely.
Thank you.
So So we can put it in the starboard bio box.
It should be, it shouldn't be too fragile.
Okay.
When you pick it up, it should bend. Will this be sample 9, May?
Yep, it's sample 9. And Jake, why don't you go for one of the 2 larger outboard bio boxes on the starboard bio box?
Sounds good.
E or F, they're both open. Did anybody see associates on that guy?
Yeah, there was a brittle star.
There's a brittle star. Nice.
Alright. Can you hit the sample salvo here? Oh, I got it. Oh, showed it. And can you switch the camera on the from the port rail to the other bio box camera?
There you go. Just let me know when you want the box open.
Yep. 1 second.
Okay, so here's a question. What happens to the samples after they've been collected?
That's a great question.
Alright, you can bump the box open.
All right. Open.
Just gonna let them focus on getting this sample secured, and then happy to talk through that.
Remind me which is one of any of the 2 big ones?
Yep, the 2
Okay.
big ones are good.
Roger. Alright, go ahead and close that box.
Closing up.
Can you bring up the craft view on the bubble camera?
Yes. No.
It's just in tight with these rocks here. There we go. Alright. Go back to
Cool.
Sweet.
There's also a sea star eating the coral I can see up in the upper.
Oh yeah.
It's really creepy when they eat. They extrude their stomachs around whatever they're trying to consume. Another fun Halloween fact. This one's definitely nomming down. You can see where it's already been, where the skeleton is bare. These cookie stars are notorious predators on octocorals, especially bamboo corals.
But yeah, to answer the question from the viewer, when these samples get back aboard, when the ROVs come back up to the surface and on deck, once everything's secured, the science team, including several of us, myself and other experts, our ocean science intern as well, we will have the wet lab set up, which is a space designed to receive physical samples, including biological samples like corals and sponges, geological samples like rocks or push cores, and water samples for eDNA filtering. So once the vehicle's on the ship, then we go out with bins and bottles and various containers to bring all of those newly collected specimens into the lab, and from there we'll process them, typically in order of biological specimens first, since we want to get them preserved as quickly as possible. And we take pictures of them, we measure them, we try to identify them on a finer resolution, maybe on a species level or at least genus level. And once they're all documented and preserved, then we will send them off at the end of the cruise to our repositories, which are like sample libraries where scientists from around the world can check out a sample and use it for their research.
If you're on Sat 3 feed, I've got the wet lab up in the bottom right corner, just so you can see what May was talking about.
Yeah, thanks Maya for that great answer and insights. I'll just add a couple more things here. So for the biological samples, we're very thankful to our partnerships with the Harvard University Museum of Comparative Zoology, who have been our repository of biological samples since 2013, have collected several thousands of specimens all over the world, many of which are species new to science. And these, as Mai pointed out, become libraries for the global scientific community to use. So any qualified researcher can request these samples on loan for them to use their, there's a form you fill out, and this museum also has fellowships, so for researchers they can apply for a fellowship, and that would provide funding for a trip to visit their museum and study the samples there. And so yeah, a lot of exploration happens here on the ship and a lot of discoveries.
Anemone.
But a lot of discoveries happen many months to years afterwards.
On the coral.
Which coral? Oh, yeah.
Yeah.
And another shout out to our geological samples repository at the University of Rhode Island, the Marine Geological Samples Lab. There's well over 10,000 samples there, and it's pretty exciting. People request them from all over the world and even work on samples collected from decades ago. So if you're a scientist tuning in from somewhere else, keep it in mind.
Right now we're looking at a Metallogorgia. It's a type of Quassigorgid coral, and in the branches there is always this snake star called Ophiocreus oedipus. They're always, every time we see one of these, there's always that snake star. And then on the stalk, there is a Venus flytrap anemone, Actinoscyphia.
And is it only this type of coral, or is it?
The Actinoscyphia will grow on pretty much anything.
Okay.
But the Ophiocreus oedipus and the Metallogorgia melanotrichus are like best buddies, and they're always together. I've never seen one without its snake star. Even when they're juveniles and not fully formed yet, they still have that that snake star in the branches. And we don't know when they get together, but at some point during their development, those two just always pair up.
Has there been studies on their some sort of symbiotic relationship, or?
Yeah, there's a paper written about it. It has a cute name. I can't remember exactly how it goes, but it's one of those little clever titles about how they're always together.
So yeah, just a little time check. So we have a little under 3 hours of bottom time to go.
I think we can do another 20 meters.
So yeah, we'll want to
Yep.
hopefully reach the summit here.
Yeah, we can keep on moving.
Definitely have quite a few fish out back.
Outside at the wire cam.
Yeah, you see them. There'll be like 4 or 5 of them swarming, and there's one coming in. Not
Cool.
sure what all they are.
Oh,
Let's take a look after watch.
Yep.
oh, on the surface?
Yeah.
Yeah.
Yeah, they'll come in like swarms when they look like they're pretty large.
Well, we usually attract a bunch of oceanic whitetips. The squids will come to the surface because of the light from the ship, and then the whitetips will come and eat the squids. It's quite fun.
Unless you're a squid, then it's not as fun.
Are
This
we,
is true.
Daniel, are we planning to be on deck at 4 or off bottom at 4?
On deck at 4. So
Okay.
yeah, it should be
Sounds
about
good.
half an hour.
I'm going to send you a DVL reset.
Send it.
Yeah, so it looks like we got about a little over 100 meters to the summit in horizontal distance. And yeah, so we'll want to try to reach that and then spend some time at the top.
Yeah, the tops of features tend to be quite interesting. Different communities will be at the top than what we see on the steep slopes. Usually lots more fish. That's especially true for geos, like flat top seamounts. The ridge seamounts can be a little more variable. So you never know until you get there.
Yeah, and this particular seamount is shallow enough to intersect with a dial vertical migrating species, so we'll see what we see up there. But it also has this really nice long ridge at the top. So I was hoping that we can make it up there and then kind of go along parallel to the edge of that cliff.
Did we collect a rock?
We did collect a rock on the previous shift.
Okay.
It ended up not being a volcanic rock, so it was a carbonate, and yeah, most of this looks like carbonate to me.
Carbonate, yeah. It just seems, we're really shallow here, so. Can we snap on that sponge, the white one?
Why not? Get a little closer.
What I'm looking for is where the stalk attaches to it.
All right, you can go for zoom.
So if it touches from the underneath and it looks like a mushroom cap, then it's Caliphacus. If it has a little osculum on top, a little hole, then it's Bolosoma. So this one looks like Caliphacus. I like saying Caliphacus. It's a good name. And there looks like there's a little amphipod on there. Glass sponges often have associates with them. That's great. Thank you.
Okay.
Bridge nav. Can we move 20 meters 015? Thank you.
While we're setting up here our next move, also just want to take a second to acknowledge our scientists ashore that are supporting us here on the chat and watching from all over the world. So we have Ikelau Oto here from Palau. We also have Asaka Matsumoto from the Chiba Institute of Technology in Japan, Emily McLaughlin from University of Alabama, James Reimer from Ryukyu's University in Japan, Jonathan Wombach in the United States, and Steve Aufskovich from the Smithsonian National Museum of Natural History. So thank you so much for joining us.
Yeah, I came up about 3 meters, but needed.
Yeah, just that wall, like,
It's a
ever
great
so
group
closer.
of people joining us. They have so much knowledge.
Absolutely. And that's always what I really appreciate and admire, that we have
It's
a
little
global audience.
brown spot. It's this
Brown
weird
spot?
rock.
oh yeah, I think so.
Yeah.
A lot of familiar faces there. Long-legged shrimp, Carcinus.
Long-legged shrimp.
I think that one is Heterocarpus. You see the red, big red shrimp?
Yeah. Now let's get a quick snap zoom.
Quick zoom there.
Yep, yeah, you're right.
Okay.
Yeah, that one's legs aren't that long. Well, I think the other shrimp's Nenomatocarcinus.
Yeah, I was gonna say.
So we're both
2 for 1.
right.
There you go.
These sponges are super weird, though.
The little ones?
Yeah.
Yeah.
Yeah, I think they're polymastia. That's
I've got more zoom if we need it. Did you want to take a closer look?
Closer look at those little sponges?
yeah, we can do a nice zoom of that sponge. I believe this is a type of demisponge called polymastia. There's also a tiny little cup coral. Got a nice good view of the Nenomatocarcinus too, which looks to have a parasite on it as well. I think it's a leech, which is strange.
Where do you see that? The
On
parasite?
on the leg of the shrimp,
Ah.
there's a little leech.
Huh.
Oh, no, I see it. Yep. Yeah.
Strange.
That must be annoying for this creature.
I don't know, maybe.
The good old ball and chain.
Yeah. Must be hard to
It's
move
definitely
around.
not the weirdest thing I've seen on one of these shrimps. Sometimes they have little isopod backpacks, which I think is really cute.
I love the anemone backpacks. Those are my favorite.
Yep,
Alright.
that is an ophiuroid, yeah.
Yeah.
Come wide, Aaron.
Yep.
Keep moving up this slope.
Beautiful view here on the
Oh,
Atalanta.
yeah. Oh, I think there's a Novodynia. See on the white coral over there, that red sea star?
Yeah, that's a spectacular view there.
Yeah.
Yeah, maybe if we can get a panoramic shot before it zoom ins.
Yeah.
Oh, yeah.
Down like that.
Would something like this be of interest for our leftover Niskin? Like this kind of
Oh, yeah, this is a nice high density area.
density? Since we have one Niskin so far, which was fired on the previous watch, which represents the bottom, and apparently they do want bottom, so when we first land, we should be getting a Niskin. So they got one of those, and then we'll be able to reach 4 other of the target depths, so then that leaves 1 spare Niskin.
Okay.
Would that be of interest to you? What do you think?
Yes. If it's possible, could we fire another Niskin?
Fire Niskin?
Oh, sure. Yeah, I'll fire an Niskin.
Thank you.
All right, let's do number 2 then. And this is sample 10.
Yeah, there should be all sorts of eDNA in here.
I know in the past we've set up on top of where we're taking an eDNA sample, but do you want me to just take it here, or?
I think here is good.
Okay.
Yeah. We're definitely on top of a lot of different corals at the moment.
Yeah. It depends which way the current's moving, but I think this is
I think it's moving
across, yeah, because they're all pointing out.
Have we
You
only,
see all the little
yeah.
particles are kind of moving across the screen.
So that's perfect. It's kind of towards us.
have we taken a Niskin yet?
Yeah, we've taken number 1, so let's put this in number 2.
Okay. Can you rack back the camera? Retract it when you get a chance?
Yes.
Hello?
All right, retracting.
Okay.
Oh.
And then can you pan left with the camera?
Oh. Yeah.
Oh, with the hydraulic.
Oh.
Yeah. The main Zeus. Sorry, I should have specified.
All right, panning left.
Thanks. Just tilt up a bit.
Go left a little more.
Maybe just hook it.
Yeah.
Seems to work the best.
Did it go? I think I saw it go. Second one?
Yep, 2 went.
Okay.
Sweet.
Megan, are you wearing a goat hoodie?
I'm a nudibranch.
Oh, you're a nudibranch. Oh, that's so cute. That's so cute.
Happy Halloween.
Happy Halloween.
All right. Is there anything we wanted to specific zoom on, or?
Oh, yeah, let's look at the brisingid on the white coral. So right in the lower of the main HD, there's a sea star there.
I think you're not on SPL. Yeah.
Okay, can go for a zoom here.
Yeah. No worries.
Oh, very nice. Yeah, I think this is a Novodynia.
Yeah.
Very cool. Lots of brittle stars too, and hydroids.
Is that a squat lobster underneath there? Or something?
Oh yeah, there's a squat lobster behind there.
All right. What was the other one we wanted to zoom on?
Just a few of the yellow ones,
Okay.
maybe.
Yeah.
I think we've got at least 3 different ones.
Yeah, there's definitely a few different species of yellow.
Okay, come wider.
There's like a fluffier one, and then there's the sort of sparser branching one.
Yeah.
There's a few different species of Corsagorgid, a Primnoid, it's a very long bamboo that's
Yep.
hanging out with us.
Trying to get around that.
There's Phreads, saw
Let's go for a
Victigorgia,
zoom on this yellow one here.
some zoanthids. Oh, there's like anemones on the rock behind it too. looks very pretty. So we have these snake stars and also amphipods as associates for this coral.
Wait, what's that like translucent on the rock face there?
Oh yeah, that's like some mucus.
Ah, because
Well, it looks like there's a little tiny hydrocoral. A stylastrid, little white.
if I come up a bit, we'll see the other yellow one.
And you see like these sort of smaller, yeah, that one's different.
Yeah?
Yeah, so we got like the fluffier one and then there's like a sparser branching yellow one right next to it.
Yeah. Sorry,
So
I'm a bit
definitely two
wobbly.
different species. This one looks more like Parasus to me. What does Asako say? Does she say anything about these?
Yeah, so she's just unsure. There's a little bit of a delay right now, but yeah, a lot of these require sclerites for distinguishing, so the gross morphological characters not being that useful.
Mm.
Okay. Come wider in.
Wonderful.
There's a really long black coral up on top here too. That one might be Parantopathes.
Yeah. Yeah, maybe let's just get that guy and then
Sure thing. Okay, go for zoom.
Looks like Parantopathes to me.
I agree. Yeah, these are some of this genus and some of the deepest ones found. It's been found in trench habitats even, not this species, but wonderful.
All right.
I think we've documented most of the things here, and maybe
There's
there's
also unbranched primnoid just off to the right, the white long stalk.
Come wider in.
There it is.
Oh, okay. Nice.
And there's another one on the rock, a really long one. So probably Candidella gigantea. Which makes it really confusing because you have two white unbranched corals, a primnoid and a bamboo. And then you have to like separate them apart. It's one of those things that's really hard to do unless you've seen a lot of them. Oh, and then there's this Peristinella. That one's cool.
Racing on by.
Lots of different types of primnoids here. There's a Furia near Oca erecta, the glass sponge. Oh. And in the Atalanta view, it looks like there's a pelagic sea cucumber. I think they're really weird. I love them. I'm gonna reset your DABL.
Roger.
Yeah, I have a feeling the densities are going to keep increasing, and then
I want to look at the fish.
just look at the topography at the top.
I think it's Iliophis.
Fish? Oh. Down here?
No, it's right up there.
Oh. Another fish.
It's a eel. Oh, it's not being helpful.
Oh. Running into its crevice.
Yeah, they do that.
Yeah, what's our priority, Daniel? To keep on moving? Get to the summit?
Yep.
Yeah,
Okay.
if we can.
Okay. Probably ready for another ship move.
Yeah, I'm ready when you're ready.
Ready.
All right. Bridge nav. We'd like to move 20 meters, 015. Thank you. There's so much stuff here.
Yeah. Could spend days here.
You could, especially when you zoom in, then there's like, oh, and there's one more thing. There's another thing.
Hey Nav, I was just wondering on HiPAC, was that last sample just not renamed, or was
Oh,
it? Oh, there.
sorry.
It just refreshed.
Yeah, sometimes you just have to move the mouse.
Okay, nice. Yeah, I saw you open the window, so I knew
I
it was
typed it
there.
in.
But
I guess I just didn't, you know,
yeah.
encourage it enough.
Wow, look at this.
Oh yeah, that's a nice one. This one's a Magnus spiralis.
Magna spiralis.
Yeah.
That's cool.
Yeah, these guys get massive, very high. I believe the tallest one of these ever measured was over 5 meters tall,
Oh my goodness.
making it the same height of an adult giraffe. It's,
Wow.
That's impressive.
All on that little stalk, too.
Mm-hmm. Oh, can we look at that crab that's on the coral?
Sure.
So, not that one, the other coral.
Yeah.
It looks like it's holding something?
This might be a good moment for a nice still from the WCA,
Oh, yeah.
if we want to focus in on that.
A bit higher. All right. Go for a zoom there.
What's it got? Oh, it's got a little piece of coral. So this is a homolid crab, a carrier crab. And they like to carry different things with them. So they have these modified back legs.
Back scratcher.
Yeah. This one's got a little piece of that yellow coral we were just looking at.
Why would it carry this piece of coral?
It likes it.
It's just fond of it.
Yeah.
good answer.
They pick different things up, you know, and every one of these crabs picks something different. So I think they have some interesting personal choice.
Is it for just like flair and
Yeah,
style, pretty
I
much?
think it's flare.
That's super cool.
Wow.
I didn't know about that.
Yeah, some species only carry one like type of an enemy, but other species will be a little more varied in their choices.
Crab style.
Crab style, yeah.
Does it normally pick up like a dead piece of coral, or would it sort
they
of
pick
break?
up live pieces. I think they might cut the pieces themselves.
They cut them. Okay, so they're harvesting their style from surrounding.
I've seen them carry pieces of sponge and different types of corals. It's usually always a live piece of something, though.
Huh.
It could be like a little bit of a distraction, like,
Come
oh,
on,
I'm
Aaron.
not a crab, I'm a coral, see? But they rarely carry the coral that they're hanging out on.
I want to see this.
It's a big Paragorgia.
Oh, there's a big dead scleractinian.
Yeah, huge.
It's too pretty here.
Yeah, it's tempting too. But trust me, when we get to the top,
Oh, it's
it'll
gonna be more interesting, yeah.
You want one cookie now or two later?
Bridge, nav. We'd like to move another 20 meters, 015.
I'm going to get out ahead of it. Kind of lagging behind.
Yeah. So I have a feeling that this cruise is going to be a fun one to annotate. It's going to take a long time.
It's a really cool Atalanta shot.
Oh, there's the
Side view.
multi-puff Caliphacus. That one's a weird one. So there's this Caliphacus that has branching, and then it has a whole bunch of these little mushroom head puffs. Ooh, Chenophore.
Where did you see this multi-puff Caliphacus?
It's like in the
Off to the-
upper, off to the left.
Oh, I see it in the other view, yeah.
Yeah. Ooh, just a nice big sea pen. The Balticina. Oh, and there's a Walteria. Lots of really, really cool things.
How are the gauges doing? I haven't looked in
Good.
a little bit. Good.
Stable. The last hour they've held solid.
There's a question here from the audience about what the substrate is, what the bottom material is, and I believe all of it is carbonate that's been carpeted with crust.
Yeah, it looks like it. The manganese crust has a kind of, well, what we call botryoidal texture. So if we zoom in, there's kind of little rounded dark texture on top of that carbonate, and all the sediment that falls down accumulates between these structures.
This dive kind of reminds me of like the musician seamounts and up in the Papahanao-Mokuakea Marine National Monument, like with the density and diversity of corals we've been seeing. So this is a very, very special place.
Yeah, since you brought it up, Megan, might be a good point to remind our viewers, our global audience, that we are in the Palau National Marine Sanctuary, this very large marine protected area that covers 80% of the exclusive economic zone of Palau, covering a wide diversity of habitats, including both the water column and the seafloor. And these large marine protected areas are really important reservoirs of biodiversity, not just protecting what we know, but also what we don't know. And Palau is world renowned for its shallow water coral reefs. We are here close to the coral triangle, the spot on the Earth where we have the highest diversity of marine organisms on Earth. But that's just in the shallow waters. In the deep waters, very few surveys have been done, and this is the first time that we really get a glimpse of what's down there. Yeah, it'd be good to get a quick zoom on those.
Yep.
Yeah, these Boltacina are really cool. I bet there's going to be a garden of these up top. See, these are types of sea pens. They root in the sediment.
Go for a zoom.
And we call them sea pens because they kind of look like feather pens. They have this long peduncle that is the pen part, sticks in the sediment. This is the feathered part with all the polyps.
A little creature coming up on the port side over there.
Oh, yeah.
Oh yeah, that looks like a fun creature.
Yeah.
Is that a holothurian?
Oh, it might be a fish.
Fish?
Is it a fish or is it a shrimp? I don't know. I can't see it very well.
Oh,
I
Oh,
think
it
it's
looks
a
like
fish.
a fish. Yeah, it looks like a fish.
Yeah. Probably a morid, a codling.
All right. If you come wide a little bit, I can track the fish.
Yeah. Oh no, it's an Oreo.
Oh, yeah.
Ah.
I like Oreos.
Just get a zoom on the Oreo.
They got cute little big eyes.
Daniel made a
Yeah.
cookie analogy earlier, so
Yeah,
it's on theme.
there were Oreos last night.
Oh, it's so cute.
Yeah, I love the little wavy spins. Just bring me joy. Nice.
Watch out for that.
It's not happy about those lasers. That's, yeah, it's better.
Yeah, in the wide angle, it looked like it was a longer fish.
All right.
Yeah, that's cool. Very fun.
Come wide, Aaron.
We'll definitely start to see more fish, I think, as we get at the top. We're almost there.
We're getting pretty close to our next targeted eDNA depth of 1,000 meters. I think we got a little ways to go, but
Yeah, not much further. Bridge, nav. Can we move 20 meters 015? Thank you.
Jake, do you have any sense for current here? Pretty still at the moment, right? Or
there's a little bit of current. I'm hands off the controls right now. Seems
Got it.
right to left. This
Yep.
orientation.
so east to west?
Yeah. Not a lot, but definitely a little bit.
Roger that.
you're trying to think about which way to go? Yeah.
Yeah, I mean,
Have we looked at the madrepora yet?
What's that?
That coral. Oh, there's a pelagic sea cucumber.
Oh, yeah.
Want to look at that, Jake?
Sure.
It doesn't really, scream sea cucumber right
Presume.
away. But yeah, this is like a very specialized sea cucumber that lives
Wow.
in the water column.
It's fitting for today.
Mm-hmm. It's giving like witch's hat.
Yeah. That's so pretty.
I think it came along for the Halloween ride.
Mm-hmm.
Nice.
Okay.
Cool.
Coming up on a coral.
That was fun.
yeah, great shot.
Yeah, the madrepora is this white one that has that, and then there's the purple stuff on
Okay.
it, which is
Is that alive or is that?
so there's a dead piece of something, and then the
Yeah.
madrepora is growing on top of it, along with the glabularia, which is the purple stuff.
Its base is really, really big. Is that?
I think it might be an old bamboo,
Okay.
is the dead part, and then stuff is growing on top of it.
Yeah, I see what you mean now. Yeah.
Go for a zoom there.
Yeah, so there's this stoloniferous octocoral, which is the purple stuff, called glabularia, and there's little amphipods on that. There's some sponge.
There's a little urchin.
Yeah, and there's a little urchin up at the top corner. And then the zigzag pattern is
Oh, yeah,
how you
there's your-
identify the madrepora. They've grown that very distinctive zigzag branching.
Given the size of the base and the thickness of the dead coral, how old do you think that would have been?
So bamboo corals have different growth rates, but probably a couple 100 years old, I'd say, for sure.
That might be a good WCA still.
Oh, yeah.
Nice.
So there's urchins, there's brittle stars, there's squat lobsters.
Yeah, and so this madrepora, those are reef building corals, so typically people think of reef building corals being in the shallows, and here in Palau there's several hundreds worldwide, in the order of 600 or more reef building corals. In the deep there's only a handful of them, and this is one of them.
Yeah, they can form pretty big mounds, and they often have a high diversity of associates. And even in this small colony, we're noticing that super high diversity of associates.
All right.
Yeah, great. Thank you,
Yep.
Jake.
Come on, Aaron.
Bridge nav. We'd like to move another 20 meters, 015.
So we're at a depth of just about 1,000, and our altitude is about 10 meters. So what do you think about the 1,000 meters? Should we wait a little bit, or since I'm not sure if it's depth or seafloor depth or Hercules depth that you're after, but it's all probably pretty similar, so.
I mean, if we can take it now. If we're gonna hover over this depth for a little bit, maybe we could wait. What do you think?
We're heading up slope, so it's just going to get shallower.
Okay, let's fire the Niskin now then.
Do the Niskin now.
Yes, please.
So number 3.
Okay. Retract. And band left.
I think 3 was already fired.
No.
Well,
Unless,
I'm seeing 3 tops.
Did I just fire it by pushing it, or
No,
you'd already seen it fired?
I think it was fired when you hit 2.
oh. Okay. Is that, are you pretty confident about that?
Yeah, because he didn't touch it yet, and I see 3 tops.
Okay, so if that's the case, then we're 1 short. So what do you think would be your preference, Makani? The high biodiversity one that we got just before this was at a depth of around 1,050 meters. So I don't know if you want to skip the 1,000 or keep it and do the other ones. It would mean that you would have two Niskins at that 1,050.
Yeah, that's fine. Maybe we could skip it, and then we'll just do the 500.
So do these 3 instead?
Yeah.
Okay. All right. So front row, we're not going to pull a Niskin here then,
Right.
if that's the case. But we do have 3 left, right?
Yeah.
Yes.
Okay, cool.
Yeah, and those will all be on the ascent.
Okay.
Yeah, we should be very close to the summit depth here. Looks like maybe a little over 40 meters in horizontal till we get to plant our flag at the top of the seamount, and then we can choose our direction. Looks like on the west side it's slightly more pronounced ridge, but we can see what we see when we get to the top.
Okay. I'm gonna try and get out ahead and come up a bit.
Should take
so
a picture with the lower camera. Look at that.
Get away from that ridge.
Yep.
For that Atlanta.
Yeah, I came up about 5 meters so
Nice.
far, but keeping an eye on her.
So yeah, looks like we should have about 2 hours of bottom time left. So excited, yeah, that we'll make the summit and then have some time to explore the top of this feature.
Did we decide which direction we're going? Or did you?
I'm leaning towards west, but we can,
You
yeah.
can see how it looks.
Yeah.
Make the call.
It looks
You
like
want to go west?
But what do you think, Megan? I mean, it's...
Well, I think east might be more gradual slope downwards, so it might be easier to survey in that direction.
Yeah, that's true.
If we go west, it's gonna be hard to look. Oh, there's another one of those multi-puffs.
Oh yeah,
Multi-puffs.
it's a better view of this one.
Yeah.
Some associates.
There's another one of those carrier crabs. It's carrying something different.
Are those, is it one organism?
Yeah, this is one organism, and it's branching, creating like multiple heads. Most of the Caliphacus that we see only have one head.
That's cool.
That one's a really pretty. I don't think I've seen one with this many heads on it.
1, 2,
It's
3,
usually
4,
like
5, 6, 7,
yeah, there's
8, 9,
usually
10.
like 3 or 4.
There's 10?
Yeah, it looks like
Yeah,
10.
10 heads. Can you come up a little bit?
It's living a very vibrant
5 meters
life.
or something?
Nice.
We have a question from one of the viewers. They are curious to know, what are your professions that allow you to be able to do this type of research, areas in the seas, and also like using ROVs?
Yeah, I can take a stab at that, but yeah, I mean, I'll just start off by saying that to do this type of work, it requires a big team, a big interdisciplinary team, engineers, natural scientists in all the different disciplines, but also educators. And here on board we have a wide diversity of people, cooks, chefs, medical professionals. And so, yeah, I think that the take-home message is, if you have a passion for this type of work, there are many different entry points. And obviously a love for the ocean, STEM background is important, but yeah.
See that little red star?
There's a lot of info about all those pathways also on the Nautilus website.
Tiny one?
Yeah, that little tiny one.
for zoom there.
Oh, that one's nice.
It's a cute one.
Yeah.
Nice.
Do you know what that one is, Megan?
Yeah, I do know what that one is, but it's eluding me. Oh my goodness.
No worries.
I was like, I gotta like get the name out of my head before we zoom in. It's oh man.
We'll check later. No worries.
All right. Come on.
It's late.
It's yeah.
Wow. It's a really big.
There's so much stuff.
You can see one of those little puff guys, but it just has one at the very end.
Oh yeah.
Yeah, so that's normal. Normally you only see the one puff with the Caliphacus. The multi-puff is different. Oh, and this big coral over off to the right is kind of neat looking.
The white one?
Yeah, that one.
Oh, yeah.
Oh, scared me. A little octans cap.
This might be a good WCA shot.
Mm-hmm.
It's really pretty. Yeah, wow.
It's beautiful.
So this is a Calogorgia, another type of primnoid coral.
Is this the Calagorgeous?
Yeah. How'd you guess?
Frame up that WCA shot.
This is a really big one. There's lots of snake stars. There's another one of those homolid crabs carrying, it looks like it's carrying a piece of like Narella or something, or another one, a different
Zoom there.
primnoid.
Yeah. Crab style.
Yeah. See, the crab's like, oh, look, I'm part of the coral. You can't even tell the difference. I'm not delicious.
Do you want to take another WCA now that we got this good view of it? I think these would be really cool pictures.
Now, it looks like these snake stars are Ophiocreus. I say they're very smooth arms, which are the same genus as the ones that are on the Metallogorgia, but this is a different species.
Do you know about the growth rate of these guys? These corals?
I think these grew a little
What do
bit
you- what do you
faster,
see,
but I
Megan?
don't know. There might be an egg, like an octopus egg sac over
Oh,
there.
yeah.
Some sort of
Oh.
thing, feature, white.
On the right, like
Yeah.
on the right side of it, yeah.
What is? I like how I'm leaning, like I'm gonna be able to see
Yeah.
that around the corner.
I think we're all doing that.
Squint your eyes
Oh, maybe
really
it's
hard.
a snail.
Yeah, it looks kind of snail-like, doesn't it? Looks like a shell
It's a big snail.
or some sort of crab.
Oh, sorry.
Huh.
Neat.
All right.
Too many good things. I often lean when I'm annotating too. I'm like, oh.
Come up on Atlanta just a little bit. The tether.
Yeah. Let's heading up.
All right, come on.
That was a cool one.
Yeah, it's a good illustration here. You can see all the sea fans all being oriented in the kind of same plane. And that gives you an idea where the predominant currents are coming from. So a lot of these fans are perpendicular to the predominant current flow. And interesting enough, on a lot of them, the polyps are actually on the downsides of the current, so they are able to get tiny plankton and other zooplankton in turbulent eddies that form on the downstream side of the current.
Oh, that's so cool. I didn't know that. Is that for most of those species, or?
Yeah, for fans, since they
Yeah.
a lot, not all of them, but yeah, when you have very strong currents that are all kind of predictable in the same direction, you have a lot of fans that are oriented. And some of the species we've been seeing on today's dive, like they're more mobile, so a lot of the the more softer, especially the spiral, like the Magnospiralis or wire corals, they are able to move with the current and kind of orient themselves
Another
depending
Oreo.
on what the best flow is.
Nice.
Those are so cute.
Yeah, they're really good at posing.
This is the same species we saw before, right?
Yeah. All right. Bridge now.
So yeah, we're very close to the summit now.
Yep.
And we should have about an hour, 45 minutes of bottom time remaining.
Hey, quick zoom here before it runs away.
Yeah. Oh, it's shiny. It could be a
Maybe it'll sit
ventrifossa.
down for us.
So this is a type of rat tail fish or grenadier. And it's really adorable.
All right.
Yeah, they're very fast, so we would be unlikely to be able to catch that.
Challenge accepted. All right, come on. I'll continue on up.
But it's not the Coelurhynchus that Jeff really wants.
How do you tell the difference?
The shape of the nose, so the Coelurhynchus kind of have these like kind of shovel noses. They're very, very distinctive. But they are rat tails as well.
Are there any questions out there from the Nautilus Live viewers?
One question is, have we found any new species on this expedition? And
That fish we captured could be new. So it's highly likely.
Isn't it quite a long process before you know whether or not something's new, or is it pretty quick?
You might know right off the bat that it's definitely something new, but it is a process to actually name it. You do have to have a specimen in hand and then describe the species and write a peer review paper.
Yeah, there's a recent paper, a meta-analysis that looked at how long it takes from the collection of a specimen to when it's properly described and a new species name is assigned to it. And on average, it's currently at 14 years.
Oh.
So a lengthy process. And this is a project and something that is really a mission statement for one of our partners, the Ocean Census Project, that is trying to accelerate the time it takes from collection to species discovery and description. We currently, when we look at bigger animals...
That fish.
Oh, pause here while we get the fish.
Looks like maybe a Nizumia. This is a different species of rat tail.
It's hiding.
Oh, yeah.
Other side of
Too
that.
shy. The bullosum was cool too.
What would be the kind of target length of time from Ocean Census's perspective for number of years?
Yeah, so it's accelerating as quickly as possible, but now one more thing for contextual information. So it's estimated that a little over 2 million species of marine animals, of the larger, so excluding bacteria and the singular-celled animals, over 2 million, and only about 10% of those have been described and named. So 90% of biodiversity still to be described. And in many cases, we're losing species quicker than we can describe it. So there's a need, obviously, to properly document things before we lose it. But it's going to take a very large effort that's going to require both developments in technology, training the next generation of taxonomists,
Get a still cam shot of this?
and also working up. So there's
that's
a lot of
gorgeous.
specimens in museums that sitting around having not yet been described. So it's going to take a all hands on deck type of approach to document the large amount of biodiversity that is yet to be found.
Bridge, Nav. Can we move 20 meters, 015? Thank you. Oh, can we check if this white one is alive? Oh, yeah, that little white one there.
Go for zoom there.
It is. Looks like a type of Pleurocorallium.
Yeah, it looked like a stylasterus when you first pointed it out, and then yeah, you zoom in, it's completely different.
Mm-hmm. That's why we gotta zoom on some of these weird ones. Like, you don't know what it is until you actually get closer and can see the polyps.
All set? That one?
Yeah. Do you think this one's worthy of a collection, Daniel?
Yeah, yeah, I mean, you know that group better than I do, so.
These little white ones aren't very well known. It could be something different.
I don't know, it's. Are we set up for Jake?
I'd have to bump over a little bit. Big yellow coral's in the way, but yeah. I can.
Have we seen any of these yet on this dive?
I don't think so.
Yeah, not on this watch, definitely, and I mean, it seems like a different fauna, so yeah, I mean, Jake, if we're set up for it, like, or it's easy enough to set up for it, let's go ahead and do that.
Yeah, all right, can you come wide a little there? Thanks.
Here is an interesting question. Have you observed much in the way of damage from pollution and climate change?
Yeah, that's a really good question. So we know of impacts, certainly, but in the deep sea, those impacts are a little bit different. So in the shallow water coral reefs, you know, there's these very pronounced impacts, but I'll just wait maybe until Jake finished this collection, and then I'll.
Okay. Are we looking at the whole specimen or just a piece of it?
You can take a piece of it. Yeah, we don't need the whole thing.
Can you go for a little bit of a zoom, a little push in there? That's good.
Yeah, no, Sako agrees with this collection.
Good. This is
This
kind
is
of
a
shallow.
stylastrate?
It's a Paragorgia.
Oh,
I got a piece of
Possibly.
it,
But that was pretty brittle, so maybe
Yeah.
it's not. Maybe it's a corallid.
so as far as boxes, I think it would be best to go in the starboard one. We do have Omega open in the forward one, but there's a floaty sample on the left, so I think we're kind of trying to avoid that box opening.
Okay.
So as far as the starboard side goes, you've got the...
Do we have suction chambers open?
We do have suction chambers open. We could do that
That
instead.
might be easier
We
since
could try
it's a
that.
small piece.
Yeah.
All right, let me see.
You can go for...
Can you, yeah, come on, thanks, Aaron. And then we're gonna tilt down.
Chamber 4 is open.
Okay, chamber 4. Let's switch to the sample salvo. Can you bring up the jar camera? Switch over to from the starboard box. So bucket, it's on that. Yeah. Nice. So that's the flush We want jar 4, right?
Sure, yeah, jar 4, 5, 6, and 7. They're
Okay.
all available, so.
So now if you can go to hydraulics page and rotate the jars until you get to jar 4, Sarah.
Yep.
I'll set up this.
All right, rotating. And we are on 4.
In any case, the family that this coral belongs to is Corallidae, because the Paragorgia are now in the Corallidae.
All right, and then
When we get in the lab, you'll be able to tell for sure. If the skeleton
ready,
is
can you
sort of
turn on
spongy,
suction?
it'll be a Paragorgia. If it's
All right, suction
or it
pump.
could be
Yep.
Subergorgia.
You can latch it on. Okay, we're seeing flow. You able to push in just a little bit, Aaron? Yeah. I can barely see the tip of it.
I can go a bit further if you want.
Oh, dropped it. Well, I'm not gonna be able to get that little piece, I don't think. Well, I can pick up this suction jar. Yeah.
Could be easier just to get the piece in front of you.
Yeah. Can you see the PRV? Can you turn it all the way up on that? Yeah.
Yep. All right. Oh.
Saw it go.
Yep.
There it is.
Yep, it's in the jar. Can turn off suction. There it is.
Steve is tuning in. Hi, Steve.
Hi, Steve.
He says that he's leaning towards Pleurocorallium for this one.
Okay, yeah, it broke really easily, so could be Pleurocorallium kishinoye. Which we wouldn't have known unless we touched it.
Yeah, excellent collection.
All right.
Yeah, it looks like we're very, very close to the summit now.
Yeah. We're almost there.
Imminent, yeah.
Yeah, we're making good time.
Yeah, I mean, I think maybe let's just take a look at the top and then, yeah, so yeah, looks like we have just under 1 hour and 30 minutes of bottom time to go. Should be reaching the summit here imminently. Yeah, from the slope, the eastern, the western side looks a little bit more interesting, but it is going to be going downwards again quite more radically. So make for it's a stunning community right there.
Yeah.
Wow. It's definitely a premium current spot, I guess.
Mm-hmm.
I like to hang out in the penthouse suite there.
Exactly.
Oh, fish.
Oh yeah.
That would be a great WCA.
Oh
When
yeah.
we move up just a little bit and get
Yeah.
the whole community, I think it'd be really cool. Cool shot.
I'll get up a little bit. Frame that up right now.
Nice. Awesome.
Yeah.
Wow.
Now, do you manually capture stills with that back in the back row?
With the WCA,
Yeah, or is it just capturing?
we at the moment manually, yeah.
Okay.
Yeah.
If we do photogrammetry, then it'll be
Yeah.
automatic, yeah.
All right.
Yeah, if we can line up like the glamour shot here.
Yeah, it's looking really good.
Yeah. Get some different perspectives.
Can we get the top end to it all?
See right through there.
Those benthic ctenophores on the chrysogorgid right in front of us.
This is cool.
Yeah, that is a spectacular view here.
So many colors.
I think in the future when we do photogrammetry, this would be a really good candidate for one of those around the boulders that Brian's interested in, to get a nice 3D model of something like this.
Yeah, no, Harold would love a 3D model of this, I'm sure.
Yeah. And Bari does a lot of that kind of stuff, like large 3D models of boulders covered in life. They're really, really neat. Oh yeah, there's a lot of these ledge features with abundant and diverse
Yeah.
colonies.
I mean, I was envisioning following along one of those.
Yeah, do we want to put in another move?
Yeah, which direction do you want us to go?
I have a slight preference for the west, but I think, yeah, you're right. It's gonna not be as good for good visuals going back down.
What's that little red guy?
It's a jelly.
Oh, cool.
Oh yeah,
Little red.
little
Zoom in on
jelly.
it.
From a nav perspective, Megan,
Yep.
you prefer east, right?
I think east would be better
Yeah.
for the ROV, yeah.
Yeah, let's go ahead and do that.
Okay. Let's do 060. Oh, that's pretty. So this is a coronate jellyfish.
Oh,
Sound good, Jake?
060, I heard? Yeah.
Bridge nav. We'd like to make a 20-meter move, 060. 060.
Another one of those multi-posts.
Yeah. And a basket star. Paracliptrophora, Bathopathes. There's a pancake urchin. Paragorgia. S1 clade bamboo corals. Vargadrella. Chrysogorgia, Metallogorgia. Parantopathes. Metacarcinus. Madrepora. Anelopsamia. Urea.
Do we know how
This little white one. Can we
these-
look at that one?
Sure.
do we know how these table-like structures form?
It gets eroded away.
Mm.
So this is all old coral
zoom.
reef that's sunken over time. And it erodes with the seawater. Oh, this is a hydrocoral, stylastrid. So this is probably a stylaster. There's a little munida on it, type of squat lobster. And there's a bunch of zoanthids and stoloniferous octocorals on the rock as well. So May was saying earlier, the black crust that we're seeing on the rock is manganese crusting. It has this botryoidal texture, which is sort of like these round, bumpy, spherical texture. Manganese coating forms over rocks in the ocean. It's a bunch of metals that precipitate out of the seawater and onto rock. It'll also precipitate onto dead coral skeletons or sponge skeletons as well.
Oh, big fish. You come
Oh,
alive?
big fish.
Yeah, and thanks, Steve, just chiming in the Silastrid is Cryptelia.
Oh, Crypthalia.
Yep.
Oh,
Oh, nice.
Oh, that's a nice big cusk eel.
Wow.
It looks so grumpy.
This is a good.
It's
Yeah,
really
nice.
posing with the big boulder of corals.
Mm-hmm.
welcome to my crib.
Yeah. It doesn't want us to see the face. I want to see your face. I don't know this one, I don't think. It kind of gets a little spectrunculus-y, but I don't know. Doesn't look right. The eyes placement on the head is different. We'll have to ask Jeff later. I think the other one that's further afield is Diplocanthopoma. That was really cool.
Yeah.
Oh, he's backing up.
Yeah.
That was cool.
There we go. Now we got the face shot.
It's another little guy.
Very pleased with our bright light.
I love it.
Wow.
So gorgeous. Look how big the pectoral fins are.
Do we have the lasers on? Do we know?
I turned them off for a second.
Just curious about the
It's
scale.
a pretty big fish.
Wow.
It looks huge.
Do you know how old these fish can get?
I
There you go.
don't, but they do live long lives. I don't know what this fish is.
It's,
It looks
its
like
eyes are
over
really
half
cool.
a meter.
Yeah, it's got really pretty eyes.
Yeah.
Would it be possible to zoom?
It's definitely a cusk eel in the family Ophidiidae.
Definitely interested in the ROV.
Yeah, and maybe a zoom on the lateral line, I think, for ID.
Yeah,
Go
that'll
for a zoom.
help.
Wow.
Nice. It's a really good shot.
This is also going to make Jeff stay.
Yeah.